Let's have a look at sequence searching and start with a BLAST similarity search. I picked the example of a specific sequence of the human hematopoietin alpha subsequence and would like to find studies discussing its expression in hamster ovary cells. Multiple input formats are allowed. The most often used is probably FASTA, and please note that numbers indicating the residue count have to be removed from the sequence. You can upload a text file, so you see this upload sequence button. And that text file may contain one or more sequences. What I'll just do, I'll paste in the sequence including the FASTA header into that window that we can see here. On the right, I set the search parameters. So I search this protein sequence directly in a protein database. A crossover search, though, would also be possible. I keep NCBI sequences checkmarked, so this database will be searched as well. And by the way, SciFinder-n will allow to retrieve up to 100,000 sequences. Under the advanced sequence search option, typical BLAST parameters can be set. For very short sequences, you can simply click this link to adjust the parameters automatically. A query coverage lower limit of 90% is the default, which I will keep for this example. A few BLAST algorithms are available. I'd recommend to keep what the system has selected. If you'd like to include gaps, you can switch to yes here. For many searches, that might be advisable to get related sequences with deletions or insertions. However, I will not activate this option for this particular example. Right next to it is the gap cost function, allowing you to define cost penalties for opening and extending gaps. The minimum match length default is 3. The default expect value is set to 10, as you can see. Please note that some searches might not yield results because of a too low expect value. So consider raising this to at least 1000 if you don't get any results. You can also switch to a different scoring matrix for the pairwise comparison of amino acids. Blossom 62 is the de facto standard, which is why it is used as the default. Finally, biologically uninteresting, low complexity regions can be excluded from the alignment with the last option on the bottom right. So exclude low complexity regions. This can be set to yes if desired. After that, we can simply start the sequence search, which will appear under the history. By default, sequence search results are sorted according to alignment identity percentage, which is the measure of the quality of the alignment, but certainly you can resort according to the expect value, query coverage, or subject coverage. Filters on the left are available, including an organism name filter, the expect value filter. There are filters available for the query coverage percentage, so the share of the query sequence in the alignment, subject coverage percentage, and that's the subject sequence and its fraction that is in the alignment at any percentage. Again, that is the quality of the alignment itself and also the sequence length. I'm interested in those sequences where the entire query is aligned to the entire target sequence. So I limit the desired sequence length to 166, since that's the length of my target. And also I want to have the full subject included in the alignment. So I go to the subject coverage and also request 100%. Answer number 1 shows the perfect match, where the query and hit sequence in the database have the same length. The purple box shows a simplified representation of the alignment between query and target sequences, indicating the length of the compared peptide chain. On the right-hand side, we can see the number of matches and mismatches. So 166 matches in an alignment of the length of 166 amino acids, so obviously this is a perfect match. The tabs below show the details of the alignment, including the BLAST score and the expect value. Subject lists identifiers such as registry numbers and NCBI identifiers, and those are linked to NCBI, so you can look up the sequence details on the NCBI page. And references show a preview of related patents and non-patent publications. Let's scroll down to the second answer. And that second answer shows a mismatch, which is visible in the graphical representation, but also in the alignment details. So in amino acid 130, we can find this mismatch, which is clearly highlighted. And then we could check the references to see the specific topic of the publications. And scrolling down further, I will be able to see more sequences with their mismatches. One mismatch is one vertical line, the graphical representation, otherwise the details are shown in the alignment tab below. Suppose I want to restrict to the perfect match only and don't want to see the other sequences in my result set. To achieve this, I can also limit the identity percentage to 100% and thus focus results to the perfect match. Now, the only one answer that we have left is the perfect match, as we've seen before. There is an option to download results to an Excel file, to also download the FASTA format of the sequence or sequences. If I'd like to have a look at the scientific papers and patents in which this particular sequence is referenced, I can click on the references icon and the system will open this. And I can see that we have obtained 258 references for this sequence. Among these are scientific papers and also patents. And since I wanted to focus on hamster cell lines as animal expression models, there is a very nice possibility of effectively limiting publications to this subject. I can scroll down a bit to go to the concepts on the left, which are the indexing expressions that our scientists add when reading through the full text of the publications, to see what the focus of the publication was. And I can click on the view all, go to the search capability, and then search for the Chinese hamster ovary cell, CHO abbreviation. See that I have different expressions related to this particular cell type. I select all on page to keep it simple and apply this limitation. Looking at the first most recent publication, we can see native CHO cell signal peptides for production of recombinant polypeptides. That seems to be pretty relevant. By clicking on the title, we can see the details. So we can have a look at the concepts that reveal what's relevant in the full text. To download this patent full text, any of the patent pack options can be clicked to see an annotated full text of this invention.